Good morning, afternoon, and evening. My name is Jorge Riera. I am Associate Professor of the Department of Biomedical Engineering at Florida International University. Today, it is a great pleasure to chair this symposium at the 20th World Congress of Psychophysiology, a symposium titled Areal, Laminar, Columnar, Origin of the event-related EEG in granular and agranular cortex of rats, monkeys, and humans. We put together a team of experts in electrophysiology, biophysical modeling, data analytics, and psychophysiology to address contemporaneous questions around the origin and interpretation of event-related potentials. In 3 years, we will celebrate the centenary of the most relevant discovery in human electroencephalography, or EEG, the Becker rhythm. There have been many substantial advances in EEG technique during the last 4 decades. In particular, after the introduction of the digital EEG recording. The non-invasive character of the EEG technique makes it very attractive to study cognition and diseases in the human brain. Epilepsy and sleep disorder were the most accepted early clinical application of EEG. The introduction of the quantitative EEG or QEEG in the 80s helps with the diagnosis and monitoring of several brain disorders. The use of EEG data in the development of brain-machine interface have been critical, given his high temporal resolution. Many groups around the world work on the development of sophisticated mathematical methods to develop a new modality of brain imaging, what is called brain source imaging or brain source analysis. This modality is used to study electrical activity from region inside the brain and have been enhanced with the development of the electromagnetencephalography or MEG in the 70s. After all these years, the origin of the EEG and MEG remains poorly understood. The representation of EEG and MEG data by cortical dipoles, cortical current dipoles, is broadly endorsed. The validity and interpretation of cortical current dipoles have been evaluated using biophysical models. The combination of EEG and MEG with invasive intracranial electrical recording have provided new insight into the neuronal origin of these segments. Creating links between these dipoles and current source density maps obtained with microelectrode array constitute a crucial experimental tool. Unfortunately, these theoretical and experimental approaches require dealing with complex problems and new challenges. Here, the level of complexity of these approaches is illustrated with few examples. Characterization of the volume conductor modeling is one of these new challenges. For example, the definition and standardization of sensor montages is mandatory to consistently record EEG from animals. In several cases, recording chambers and conductive head fixation devices are used, which need to be incorporated in the forward mode. The scalp of these animals suffers deformation due to surgical procedures, and in some cases, EEG electrodes are anchored in the scalp. To deal with these practical issues, either boundary element method or finite element method need to be used or have been developed. How different cortical neurons contribute to EEG is another important question. For example, it is well known that layer 5 parameter cells comprise highly dense L-type calcium channels in the proximal apical dendrite and trunk. This calcium channel help these cells amplify distal synaptic input by means of calcium spike. We have recently developed a simplified model for layer 5 parameter cell that allowed us to evaluate the local field potential and its related current source density distribution generated by both action potential and calcium spikes. Using this model, we have evaluated the errors made by simplifying thousands of these neurons in a cortical column by a single dipole for different species. Our analysis revealed that the accuracy of the dipolar representation depends on the relative dimension of the head with respect to the sensor distribution. Increases in the error were more pronounced for EEG than for MEG. The error were more substantial in the dipolar representation, were more substantial in animals with small heads, and is more accepted for human and monkey, but not for rat. However, layer 3 parameter cells do not have a calcium-mediated facilitation, producing a different local field potential and consequently a different CSD distribution when substantially activated. They are then dendrite with synaptic inputs. The definition of the center of the dipole is another relevant aspect to consider when calculating the time-varying amplitude from the CSD spatio-temporal map. On the left panel, current map are directly calculated from the transmembrane current of thousands of neurons of a layer 5 parameter cell after calcium spike and action potential were evoked by synaptic stimulation. The indirect calculation of this from the CSD map of the CSD map from the simulated local field potential is shown on the right, which reveal a very similar spatiotemporal panels. The respective dipolar amplitude are shown on the lower panel, which are calculated with different formulation. However, the amplitude of this dipole is very sensitive to the selection of its center. Here we illustrate this by calculating the amplitude of the dipole, selecting the center of the dipole in the center of mass of the column, or when it's selected in the center of charge, represented here with dashed lines. The origin of oscillatory activity commonly observed in experimental paradigms has also been a subject of debate. The panels on the left show the presence of theta and gamma oscillation evoked by saccade edge movement during a contour-minded task in monkeys. Most accepted proposals for the origin of this ring are based on cortical circuital properties. For example, fast synchronization in gap-connected GABAergic interneuron have been proposed to underlie gamma oscillation. And more recently, closed-loop feedback among parameter cells have been suggested to support theta oscillation. However, Recent simulation by our group have shown that theta rhythmicity in the order of 5 to 8 Hz could be an intrinsic property of individual layer 5 parameter cells if their apical dendrite are randomly stimulated using AMPA and NMDA synaptic inputs, a property that is not conserved if the stimulation occurs at the level of the basal dendrite. For now, we will leave this fundamental problem for future work. This symposium attempts to propose solutions to several other important questions, which are: how do multiple cortical areas contribute to an EEG and MEG component? What are factors in the line and mismatch between EEG and CSD? How do a spike and local potential contribute to the CSD? What difference do a slower ionic diffusion make on CSD estimation? Are biophysical models from granular cortical areas canonical? How does oscillatory activity mesh as EEG and MEG biomarker? Although we will not cover all these questions in this symposium, the invited speaker will try to address many of them. Thank you very much.
Hello, I'm Gert Einroll. It's a great pleasure to participate in this very nice symposium. And what I want to talk about today is how to model EEG signals from the bottom up. I mean, there are now many different electrical measurements that you can do to measure brain activity. For example, you can insert an electrode into the cortex and then pick up both spikes and local field potentials, LFPs, from the surrounding neurons. Or you can put the electrode at the cortical surface, do what's called ECOG recordings, where you can measure activities from the same neurons. Because these measures are invasive, so you never do this on humans unless there's a clinical need for it. However, you can do non-invasive recordings, such as EEG, where you measure the electrical potential at the scalp surface, or even measure magnetic fields just outside the brain. So our group has done a lot of work in the past on how to model and analyze local field potentials, that is, the potentials recorded inside the cortex. And here, the basic formula relating activity in neurons and what you measure is shown here. So if you have sort of like this multi-compartmental neuron model that has like many compartments labeled N, then if you know all the transmembrane currents in all these compartments, you can compute the potential at any position just by using this simple forward modeling formula. And there's one material parameter here. It's sigma, and this is the extracellular conductivity, which tells how easily essentially the electrical signal propagates in this neural tissue. And this formula assumes that there's a constant extracellular conductivity. And this turns out is a good approximation when you're inside the cortex, like for LFP or spike recordings. Now we have also pursued this whole idea or this scheme to also to look into the forward modeling of EEG and also MEG signals. A complication then is that sigma is no longer constant. If you sort of want to model the EEG signals, that's signals recorded at the top of the scalp, there's a lot of the layers or different parts of the head has that through which the signal has to propagate has different connectivities. So you need a model, you need a head model. One model which is popular and has been, at least we like to use it, is this idealized spherical head model. Of course, the geometry is very simplified, but it has these key features of these different layers with different electrical connectivities. This allows for computing actually getting some analytical formulas for what the EEG signals look like, well, given how it depends on the underlying neural activity. But you can also do a more detailed head model and record sort of using MRI measurements and make sort of like these sort of person-specific electrical head models. And as it turns out, if you want to link the sort of when you want to link the underlying neural activity and do like network modeling and compute these EEG signals, it's not a very big complication to look at, to consider detailed head models if you're interested. So the force sphere head model shown here, it takes into account that for a signal that has to propagate from the inside the cortex, that's shown in white here. This is a P, represents the current dipole moment from, say, a single neuron. And it has to propagate through this blue thing, the CSF, with a high connectivity, the skull, that's the bone, so it has very low connectivity. And then it's a scalp, which has about the same connectivity as the brain tissue. So we'll consider that model a lot in our or in some examples shown here, and just show some results for this detailed head models at the end. So if you want to do EEG calculations, there are 3 source models you can use, and this is illustrated by this sketch here, this cartoon here, where we have a 3-compartment neural model. So we have one you have a one soma compartment, and you have 2 dendritic compartments. And in this case, we consider if you have a excitatory synaptic input onto the soma, you get a current going in, it's called a sink, and then this current has to leave either at the first dendritic compartment or this second dendritic compartment. And then there are 3 ways to compute the extracellular potential. You can either, in this case, we actually neglect the problem with say varying extracellular connectivity, just to illustrate the point. You can do a compartmental-based model. That's the same thing as you do for the local field potential, and just keep track of if you know all the transmembrane currents in these 3 compartments, you can just use this simple formula. But there's also an alternative view. Instead of computing the transmembrane currents, you can instead keep track of the axial currents going between the compartments, because each of these axial sort of intercellular currents sets up this current dipole, small current dipole, the current going between 2 compartments. So in this case, there will be 2 current dipoles, one from the soma compartment to the first dendritic compartment, and the other from the first dendritic compartment to the second dendritic compartment. And then you can use another formula, this dipole formula, and sum over contributions from all these dipoles. Or you can do a more dramatic approximation and just assume a single dipole. Then and then you have this simpler formula. You don't sum over anything, but in order to get this current, in order to get this single dipole, you have to sum over do a vector sum essentially over all these dipoles set up by these different currents between the different compartments. So, and we can already see here maybe that hints at what will be a take-home message is that far away from the neuron, all these 3 source models give essentially the same result. It's only when you're close to the neuron that they deviate. And if you're then only interested in the potential far away from the neuron, then you might as well use the simplest single dipole approximation. So now we're going to test it. So we now want to look at a more advanced neuron model. This is still a single neuron, but it has all these compartments. So then we can compute it either with the multi-compartmental formula, or divide it up into all these little dipoles between the compartments. or do the single dipole approximation. And one thing we see is that the compartment-based and multi-dipole approximation essentially gives the same results, even close to the neuron. The single dipole approximation does not give the same result close to the neuron, but as we can see down here, far away from the neuron, now we have changed the length scale, then we can see that they essentially give the same results. So the take-home message is that the single dipole approximation is excellent far away from the neuron. We also see that, well, the multi-dipole actually gives perfect results because then the dipoles are so small that that you're actually far away from, in terms of compare the distance from the neuron to the size of these tiny dipoles, it's still quite large. So now we're going to look at so how far away do you have to be from a neuron so that the EEG, the single dipole approximation, is applicable? Well, we still consider we still know a single neuron, and here we have two situations. We have put a neuron on just at the top of the cortex, and then we have one situation where you have a excitatory synaptic input up here, and that's sort of the green curve. And then you have another situation where you have excitatory synaptic input here close to the soma. And if we just first focus on the green things here, we see that the dashed lines are multi-dipole. So that essentially means that is the ground truth. That is the that is sort of like what the computer signal should be. And we see that this single dipole approximation, the solid line, works well when you're far away, at least out in the EEG layer here. If you start going inwards, you see that it's still okay inside the skull if you're able to measure the signal there. But if you get even closer and down to the cortical surface, then we can start seeing some deviation. And more or less the same thing is seen for this example with the basal input, even though the errors are somewhat larger. However, this is a situation where the signal itself is much smaller. So when there's like many synapses contributing, many neurons contributing to the EEG signal, probably these synapses can contribute much. So the error will play less of a role. So I would say that the take-home message is that the single dipole approximation is excellent if you want to compute EEG, but maybe not if you want to compute the ECOG. Of course, it has to do with how accurate you need the result to be. One thing that we have learned is that the EEG potential from a single neuron, and also then a collection of neurons, really depends strongly on where the synaptic inputs are. That's illustrated in this plot, where we have three different morphologies. But what really matters mostly, like qualitatively for what EEG signal you get, is whether there's like apical input, in this case basal input, or uniform, and so on. And the corresponding results are shown down here, and we see that the EEG is very much different, very different for these different situations, different synaptic inputs. But they sort of directly reflect the current dipole moment of the single current dipole moment from this, like resulting from this stimulations. So we can see here that the single dipole and the ground truth multi-dipole formulas give essentially the same result. So here's an example where we compute the LFP EEG and also the MEG signal from 10,000 biophysically detailed neuron models. That's shown here. The synaptic input vary over time in this little animation, and we can see that both the LFP measured in the middle of this bush of neurons, and also the EEG and MEG contributions really vary depending on where the synaptic inputs are, and for the case of EEG and MEG, also with the orientation of the population. I mean, the key take-home message here is really that, well, if you have a network model like this one, or any network model, we can then, using these tools, LFP and these schemes that I just described, you can compute not only LFP and spikes that also fall from the same formalism, but also EEG, as well as MEG. MEG, of course, has a different forward modeling formula, but again, it's, if you know the current dipole moment, you can compute the MEG. So here's just an example where we compute LFPs and EEGs and fallen from point neuron networks. So point neuron networks are popular, that integrated fire type neuron networks, because they are easier to deal with conceptually, but they requires less computer power, so you can run them more easily on normal computers or for longer times. So, but the problem is, if you want to, you can sort of use them to compute spikes, because membrane potentials are produced in these models. However, extracellular potentials are not, because you need at least to have 2 compartments in your model to have any kind of extracellular potential. So point neuron model doesn't really have an extracellular potential. But then we have used this trick where we first compute the spikes. This is the case for this Potjans-Diesmann model, 80,000 integrated fire neurons mimicking populations in the visual cortex, and then retrieving thalamic input at least at time 900. Then you can compute these spikes. These are just some raster plots from neurons in these different populations. And then what we did, we stored these spikes, and then we replaced each of these 80,000 point neurons with multi-compartmental neurons, and different multi-compartmental neurons depending on where in the cortex the point neuron is. And then we can just compute the LFP, and also what you would measure, like in this example, EEG with a single electrode at the top of the head using the force field model. So we see at 900, when there's like a strong volume of activity in the terms of spikes, and because of the thalamic input, we can then compute all the spikes, the LFP and EEG. So that means if you have sort of like a candidate model, you think, oh, this is how you have a candidate model for how the brain, like how a piece of cortex works. Then you can compute all these different measurement modalities, and then you have more, and compared with these different experiments at the same time, and have more ways to constrain your model. And the model here means that every time you change the parameter in your whatever connectivity, that means that it's a new model. So this is a way to have like more, at least for me, it's an important motivation to be able to constrain networks models by more means than just using the spikes. And one simplifying feature that comes out of this analysis is that you don't really need a single neuron dipole either. You can have a population dipole. So the net current dipoles for like these populations of all, these populations. So you, in this case, I think you only needed if you have a current dipole for each population, that means that there's 8 current dipoles you need, and then you can compute the EEG and MEG just by knowing these 8 different current dipole moments. And this decoupling, that means that if you can use the neural network simulators to compute the current dipoles, and then you can plug that into a head model you want. Like in this case, we have considered the force field model, and get, compute EEG, for example, at the with using that head model. But we also show here that if we can take the same current dipole that we computed from this network and sort of play around and place it inside different lobes in this, using a reconstructed, more detailed electrical head models, like the New York head models shown here. So that is what I plan to say. So thanks a lot for your attention.
Hi everyone, I'm Stephanie Jones from Brown University, and I'm thrilled to be part of this symposium and to have the opportunity to describe our software tool, Human Neocortical Neurosolver, or HNN. and how it can be used to study the mechanisms and meaning of event-related EEG signals. So I'd like to begin with an overview of HNN. The core of HNN is a computational neural model of a canonical neocortical column that's designed to connect macroscale human EEG signals to the underlying cell and circuit level generation. And in doing so, provide a translational bridge between these powerful human signals and the wealth of data and techniques that are being applied in animal models, such as mice and monkeys, to study human brain processes in health and disease. And so this type of neuromodeling is a primary focus of my lab. And we've recently turned this modeling framework into a user-friendly software tool for the community to begin to develop and test hypotheses on the circuit origin of their human signals. And so my talk, I'll begin by giving you some of the background on the computational framework used in the development of HNN. And then I'll go through an example application of HNN based on studies in my lab aimed at uncovering the circuit mechanisms involved in tactile perception. I'll describe how HNN has been applied to study the mechanisms underlying somatosensory event-related potentials, and also the influence of spontaneous brain rhythms on correlates of perception in these evoked responses. And so how do we begin to develop a framework that can connect these extracranial measured signals to the underlying cell and circuit level generation? Well, we first have to understand where these signals come from. And in practice, the first thing we do is we take the extracranially measured signal and we apply inverse solution methods to estimate the location, the direction, and the time course of the underlying big electrical currents in the brain, known as primary currents, that create the recorded sensor data. From there, we have to think about the neural mechanisms that create these primary currents. And the primary currents have been shown to be generated by the postsynaptic intracellular current flow in the long and spatially aligned cortical pyramidal neuron dendrites. Essentially, the length and parallel alignment of these dendrites enables a big electrical current to be generated that can be recorded outside of the head with MEG and EEG. So knowing that this is where the signal comes from, there are some key canonical features of neocortical circuitry to take into consideration when developing a model to study the circuit level generation of these signals. The first is that the cortex is a layered structure. It contains various types of synaptically coupled excitatory inhibitory neurons in the supragranular and infragranular layers, including these pyramidal neurons with long apical dendrites. Second, these local cortical circuits don't sit in isolation, but they're constantly receiving input from other parts of the brain. And there are two primary pathways of information flow into the cortex. One input comes from the lemniscal thalamus, which relays sensory information through the thalamus and up to the granular layers of cortex, where it then propagates to the other layers and effectively hits the proximal dendrites of these large pyramidal neurons. The other input is from higher order cortex, or non-specific thalamic nuclei that project right up to the supragranular layers and effectively hit the distal dendrites of these pyramidal neurons. Now, both of these types of input form excitatory synapses on the pyramidal and interneuron populations, and they drive activation of the network. And then primary current dipole signal comes from the intracellular current flow across the entire population of pyramidal neurons. And the units of this current flow are current over distance, or nanoampere meter. And with this level of construction in the model, we have one-to-one correspondence between the output of our model and our source localized signal, which is also in nanoampere meter. And this is the level of detail that's included in HNN, but this is a reduced schematic. The full network contains a scalable number of neurons and represents a patch of cortex in a localized brain area. And we've been specifically designing HNN to study some of the most commonly measured signals, including ERPs and low frequency brain rhythms. Now, this is a very complicated large-scale network model, and it can be difficult to work with if you don't have any experience in neuromodeling. And so to make this tool accessible to a broad MEG and EEG user community, we've embedded this model in a user-friendly graphical user interface. We've created a website that contains installation instructions, and we also provide tutorials on how to begin to simulate ERPs and low frequency brain rhythms. And these tutorials build from our own prior published studies, and we give example data sets and parameter sets to get started with. We're also working hard to follow best practices in open source software design, and all of our code is distributed on GitHub. So now moving to an example application of HNN to study the circuit mechanisms involved in tactile perception. So in my lab, we perform tactile detection experiments where we give a subject a brief tap to the middle finger and ask them, did you feel it or didn't you feel it? And we record the brain activity with either EEG or MEG and use inverse solution techniques to isolate the contribution from the hand area of S1. Here's what the S1 evoked response looks like from a fairly strong tap to the fingertip, and we've loaded this ERP into our HNN software. Now, the first application of this model was to use it to understand how this ERP is generated. And so I'm going to walk you through how we did that. The first thing we did is we went to the literature. And there is a long history of literature investigating the origin of the somatosensory evoked response. And this literature suggests that the ERP is generated from a specific sequence of activation of the local S1 circuit that consists of a thalamocortical feed-forward input at approximately 25 milliseconds, that's followed by a feedback input at approximately 70 milliseconds, followed by a re-emergent feed-forward thalamocortical input at 125 milliseconds. And so in HNN, we can test the hypothesis that this sequence of activation can generate this human S1 ERP. And to do that, we click on the Set Parameters button, and this will open a dialog box that allows us to specify the parameters in our network, including the parameters that define these inputs to the network, which here we're calling evoked inputs. What I'm going to do here is I'm going to load these inputs in from a parameter set that's provided with the software. And when I do that, what you see at the top of the GUI is a histogram of the action potentials that provide these driving inputs. Now, when we hit run simulation, these trains of action potentials are going to activate the network and create a response that can be read out in the current dipole signal. So now when I hit run simulation, the network is activated, and you can see that this sequence of drive is able to accurately reproduce a recorded S1 ERP. Now, importantly, the literature guided us as to what this sequence of input should be, but we spent a lot of time tuning the timing and strength of these inputs to get an accurate representation of this signal. But our tool is designed to connect to the microcircuit activity, so you can go in and you can view various components of the underlying microcircuit. And here I'm showing you some examples where you can look at the somatic voltage of the different cells in the network. You can look at the layer-specific dipole responses, or you can look at the profile of spiking activity from all of the different cell populations in the network. And this detailed data provides several benchmarks over which you can begin to test the model predictions with invasive recordings or other imaging modalities. I've shown you some examples from S1, but HNN's model is based on canonical features of neocortical circuitry, and as such, it can be used to study signals in other brain areas, and particularly in primary sensory areas. So what I'm showing you here is source localized evoked responses in primary auditory cortex from the brief presentation of a sound, and also in primary visual cortex from a brief flash of a checkerboard. And you can see that there are some differences in these evoked responses, but there's also striking similarity, and that the signals go up, then there's a prominent negative deflection, and then the signals go up again. And what we can show in HNN is that that same sequence of input, consisting of feed-forward, feedback, feed-forward input, is able to reproduce these common evoked response signatures in these different primary sensory areas. And this consistency in the mechanisms that create these ERPs suggests that the macroscale observables are constrained by commonalities in the canonical structure and input patterns of the neocortex. From here, we can use HNN to investigate the neural origin of changes in the ERP across experimental conditions. We have several published examples where we've applied HNN for this. For example, we've looked at the circuit origin of hemispheric differences in auditory evoked response potentials. We've looked at the impact of non-invasive brain stimulation on somatosensory ERPs and also changes in these ERPs with aging. And there's a full list of publications on our website. Now going back to our tactile detection study, we've been looking at differences in the evoked response on trials where subjects reported they felt or didn't feel that tap to the finger. And what I'm going to show you next is that the ongoing pre-stimulus state in the brain before you tap the finger predicts whether the subject will feel that tap to the finger. And H&N can help us understand why. And so here what I'm showing you is the frequency transformation of our time domain response. And you can see clearly that before we tap the finger, there's an oscillation that has high power in the 15 to 29 hertz or so-called beta band. And this power changes at any moment in time, and we've actually shown that it shifts with attention. And so we wanted to ask the question, does this pre-stimulus beta rhythm influence the ability to feel that tap to the finger? And the answer to that question is yes. Here I'm showing you the detection rate as a function of average pre-stimulus power that's been sorted from low power to high power. And you can see that when you have higher pre-stimulus power, you're less likely to feel that tap to the finger. Beta power also has a direct influence on correlates of detection in the evoked response. Here, the light curves are the evoked response averaged over detected or non-detected tactile inputs. You can see that there's differences in the amplitude beginning at approximately 80 milliseconds. Now, similar differences emerge if we instead average over trials that had low pre-stimulus beta power or high pre-stimulus beta power, suggesting that there's a causal relationship between pre-stimulus beta power and detectability of the tactile stimulus that's reflected in differences in the evoked response. We next ask, can H&N help us understand why there is this direct causal relationship? To address this question, we're next going to use H&N to simulate these beta rhythms. And then we can ask, how do these beta mechanisms cause the ERP to be smaller? And when that happens, why does the subject report that they didn't feel the tap to the finger? So next we're going to simulate the beta rhythm. But before we can do that, we have to look at our data more closely and understand what these beta rhythms look like in more detail. Now, everything that I showed you before in the frequency domain was on average data. Here I'm showing you the unaveraged signal. And when we look at the frequency domain and non-average data, what we see is that these beta oscillations emerge transiently. They typically last less than 150 milliseconds, and we refer to them as beta events. They also have a very stereotypical waveform shape that's defined by this prominent negative deflection that lasts 50 milliseconds. This is 50 high power beta events from one subject, and this is the average and standard deviation across subjects. And so next we wanted to apply H&N to understand how is this waveform shape of these beta events generated. And due to time constraints, I'm just going to give you the punchline of our study. Through a lot of trial and error in trying to match the output of our model with this waveform shape, the model suggested that these brief beta events emerge from the integration of a broad proximal excitatory synaptic drive that's simultaneous with a strong distal drive that lasts 50 milliseconds. And what happens in the model is this proximal drive comes in and it pushes current flow up the dendrites. At the same time, this distal drive comes in and it pushes current flow down the dendrites for 50 milliseconds, and we get our beta event waveform shape. Now, with this mechanism, the model, we were very nicely able to reproduce various features in our recorded MEG data. And due to the level of detail, we could also test these model predictions in different domains. And we've been fortunate through collaboration to be able to validate some of these model predictions with laminar current source density recordings in both mice and monkeys, and now also with laminar resolution MEG in humans. Okay, so we're able to simulate these beta events. We're able to simulate the evoked response. Can we now combine those two things and get a prediction from our model on how the mechanisms creating this beta event have a direct influence on the evoked response? And the answer is yes. When we simulate an evoked response with and without a beta event in our model, the model reproduces these differences in the ERP that we see on detected and non-detected trials in our MEG data. And so we can look more closely at what's going on in the model. And what the model shows is the following. Recall that these beta events are generated by a strong distal drive. This distal drive is not only exciting these distal dendrites to push current flow down, but it's also recruiting inhibition in the supragranular layers. If you have a lot of inhibition in the supragranular layers and you tap that finger and a feedback input's going to come in to create this peak in the ERP, that feedback is shunted. The input can't come in because we have a lot of inhibition. You get less current flow down the dendrites, and that creates less spiking in these pyramidal neurons. If you have less spiking in these pyramidal neurons, which are the output neurons of that primary cortical circuit, then you have a decrease in the relay of information out of S1, and the subject says, I didn't feel the tap to the finger. Now we can look more closely in the model, and what I'm showing you here is the spiking histogram in the different cell populations, the excitatory and inhibitory cells, when I simulate an evoked response with and without a pre-stimulus beta event. If we have a pre-stimulus beta event, we drive a lot of layer 2-3 inhibition, and this shuts down all the rest of the spiking activity. When we don't, we get robust spiking in our cell populations. We infer that their information is relayed out of S1, and the subject says, yes, indeed, I felt that tap to the finger. So in conclusion, HNN simulates the primary currents underlying EEG and MEG signals with cell and circuit level detail in a neocortical column model. It can be applied to develop and test hypotheses on the neural origin of sensory evoked responses in low frequency brain rhythms. And it's led to novel predictions on the circuit origin of these transient beta events, and suggests that these beta events are decreasing the salience of sensory input via the recruitment of supragranular inhibition. Hopefully I've convinced you that HNN can be applied to uncover circuit mechanisms underlying EEG and MEG correlates of both healthy and neuropathological states within a user-friendly graphical user interface that's designed for accessibility by the broad MEG and EEG community. And with that, I just want to end by thanking you all for your time and thanking my collaborators and funding sources listed here. A huge thank you to the HNN development team. This is a multi-institutional team with an amazing collection of collaborators, and what I've shown you today wouldn't be possible without each and every one of them. So thank you all for your time.
I'm Mike Cohen. I'm an associate professor at the Donders Institute for Medical Neuroscience in the Netherlands. And I'm going to present a method, this is a novel algorithm for identifying empirical frequency boundaries in multi-channel electrophysiology data, and therefore in cross-scale data sets. I am very interested in trying to understand the nature of the dynamics across different scales in brain activity, across different temporal scales and across different spatial scales. It turns out that cross-scale interaction research, so doing this kind of research, is quite challenging. It's quite difficult. There are many reasons why it is difficult, and I'm going to outline a few of the major challenges of implementing successful cross-scale interaction research before presenting the novel algorithm that I think will address at least some of these challenges. One challenge is in simply acquiring data simultaneously across multiple spatial scales. So something like recording EEG and LFP and spikes simultaneously. This is certainly not impossible. There are many great labs around the world who are beginning to collect these kinds of data, but it is still quite difficult. It is technically demanding. And most research labs, certainly not all, but most neuroscience research labs are focused specifically on one spatial scale. So for example, dendrites, or only measuring EEG or something like that. But fortunately, the technology is improving rapidly, and this is allowing us to record more and more data simultaneously from the brain. For example, here is a plot showing the number of simultaneously recorded neurons, so basically a measure of the data set size, the multivariate data set size, and you can see this appears to double about every 7.5 years or so over the past half century. So collecting cross-scale data is challenging, but this is getting better over time. Now, we are also lacking some really great theories, really specific theories that will help us know what to predict and how to interpret our results of cross-scale interaction data analyses. Now, neuroscience is not entirely an empirical science, but it is dominated by empirical research, and that's because, we still have a lot more to learn about how the brain works before we can get to a level of theoretical sophistication that exists, for example, in physics. Fortunately, though, we are also improving in this aspect, and by we, I mean the neuroscience community in general. So there is increasingly some very detailed work, including this very beautiful paper that came out recently, that is helping us work towards mathematically defined theories and therefore better falsifiable hypotheses that we can then empirically test in data. Another challenge of cross-scale interaction research is the data analysis methods. Now here the issue is that most of the analyses, the statistical and data analysis methods that are used in neuroscience come from a long history of traditional univariate analyses. Now, traditional univariate analyses are fine, but arguably they are not suitable for the kinds of large-scale multivariate and multi-scale, so really high-dimensional data sets that we are beginning to acquire. There are several reasons for this claim. One is that mass univariate and also even bivariate analyses, they tend to run into statistical issues like multiple comparisons issues, for example. Secondly, to the extent that neural computations are distributed across different neurons, across different circuits in the brain, then recording data and focusing the analyses on individual neurons or individual electrodes or individual voxels or pixels may lead to poor SNR and poor estimations of the actual underlying neural computations, assuming those computations are spatially distributed, which is something we increasingly believe in neuroscience. Another source of challenges in cross-scale interaction research is variability of oscillations, and this is something that interests me in particular, and this is the primary motivation for the algorithm that I will present in the rest of my talk. So neural oscillations are a very prominent feature of the data. They're also a very useful foothold of the data, of neuroscience data, for linking dynamics across multiple spatial and temporal scales. So here you see a plot showing power spectra. So this is frequency on the x-axis, power on the y-axis. The black thick line here corresponds to the average from one particular electrode over a group of individuals. But when you look at the individual power spectra, you see that there is a lot of variability. So not only variability in terms of power, but also in terms of the frequency, the peak frequency, and the bandwidth of these oscillations. Now, mostly in the neuroscience literature, people select a frequency range typically based on integers, so 8 to 12 hertz, for example, and just use that common window as a frequency range for every single individual. I think, however, this variability should be leveraged. We should try to take advantage of this variability over different subjects. And then the final challenge of doing cross-scale research is that there are many sources of noise in neural data, and the characteristics of the noise may depend on the spatial scale. Fortunately, however, multivariate data analysis methods allow us to leverage weak correlational patterns across many different channels in order to enhance the signal-to-noise ratio. And here you see a simple example of this. This is with simulated data, so we know what the ground truth is, and this shows the distribution of simulated 10 Hz activity. So this is from the data simulation, and this shows the power at the electrode level. Now you don't really see a good reconstruction due to excessive noise, and that's the black power spectrum. You barely see a peak in the, around 10 Hz over here. However, by applying special multivariate techniques, we can leverage the correlational structure across the different simulated EEG electrodes to vastly improve the signal-to-noise ratio and discover a feature of the data that is present but might be difficult to observe when only plotting the results from an individual data channel. So this really relies on a technique called spatial filtering for trying to do source separation or unmixing sources. Again, the idea is that there are patterns of correlations that are distributed across multiple EEG electrodes, although the methods I will present are general. They can be applied to any spatial scale, or really any kind of time series data, multivariate time series data, where oscillations can be expected. And so we take all of the data from the individual channels, and we compute the weighted sum over all of the channels, and the idea is that the component that results from this weighted combination of the data channels highlights some specific feature of the data that is distributed across all of the electrodes and is therefore only very weak at individual electrodes, but can be much more robust when we consider the weighted average over all of the electrodes. Okay, so the goals of this study were to develop an automatic algorithm that can identify empirical frequency boundaries based on covariance structures, narrowband covariance structures in multi-channel data. After explaining how the algorithm works, then I'm going to show you some validations in simulated EEG data, and then I'm going to show you an application of this algorithm to a publicly available data set, and that is some resting state EEG data in Parkinson's patients. Okay, so now I'm going to walk you through how this algorithm works. I call this GED bounds. This stands for Generalized Eigendecomposition Based Boundaries. So the first step of this algorithm is to create two covariance matrices from the data. One covariance matrix is based on the broadband data, so non-temporally filtered data, and another covariance matrix is based on narrowband filtered EEG data. So you see here, this is a depiction of broadband data, so X would be the data matrix. We create a channel by channel covariance matrix. Here is exactly the same data. So this data matrix is taken from the same individual, the same electrodes, the same time windows, the same task. Everything is the same. It's just that this is non-temporally filtered and this is narrowband filtered at a particular frequency. So then we get our two covariance matrices here. The next step is to compute something called a generalized eigendecomposition on these two covariance matrices. Now, in a few moments, I'm going to explain why generalized eigendecomposition is the right thing to do here. I'm going to justify that analysis choice. But for now, suffice it to say that the result of the generalized eigendecomposition is a set of eigenvectors and corresponding eigenvalues, and the interpretation of these eigenvectors is that they maximally separate the covariance matrix S from covariance matrix R. In other words, the eigenvector associated with the largest eigenvalue will isolate a pattern, a spatial pattern in the data, such that the weighted combination of data channels maximizes the energy or the power of the narrowband filtered signal compared to the broadband filtered signal. Okay, so we take that eigenvector associated with the largest eigenvalue, and we store that for one frequency, for example, 4 Hertz. And then we repeat this procedure where this covariance matrix is the same for all these frequencies, and this narrowband filtered covariance matrix is different, and then you can select whichever frequency ranges you like. In this case, I went from 2 Hertz up to 80 Hertz. Now, once you get this collection of eigenvectors over all of these different frequencies, you correlate them. So this is a matrix of all of the correlations between the eigenvectors at different frequencies. So just to be clear, you repeat this for many frequencies, then this pixel here, the color value at this pixel, is the correlation between the eigenvector at 20 Hertz and the eigenvector at 6.8 Hertz, or whatever these frequencies are. So then you can see this correlation matrix here. Remarkably, the correlation matrix becomes organized into a block diagonal-like structure. And the interpretation is that all of these dark values here are highly correlated eigenvectors. So that means that the spatial temporal characteristics of the multi-channel data are very similar in this frequency range, and these frequencies are collectively very dissimilar from all the other frequencies that I extracted here. So after identifying or computing this correlation matrix as eigenvectors correlation matrix, the next step is to apply a clustering method called dbScan that will identify the clusters in this matrix, and that you see outlined in pink here. So these pink boxes are the result of the dbScan clustering. So now each of these clusters corresponds to an empirical frequency, and this is great because we do not need to specify a priori the boundaries of the frequency bands. The frequency boundaries are just extracted automatically through this procedure. Okay, and so that's the algorithm in brief. What I'm going to do now is take a moment to explain why a generalized eigendecomposition is the right thing to do. And that's also going to help you understand why this algorithm is useful. So our objective here, our goal, is to identify a spatial filter in W, so a vector of filters for each data channel, such that the weighted combination of data channels, which gives us a component time series, has more energy for the narrowband signal relative to the broadband signal. So this would be the variance or the norm of the narrowband signal, and this is the variance or the norm of the broadband signal. So we want to choose a vector W that maximizes this ratio here. So how do we go about solving that? Well, we start by rewriting this term as the dot product of this vector matrix product with itself. So that looks like this. And then we apply a little bit of standard linear algebra, and we end up with W transpose. And then the key terms here are in the middle, X times X transpose. Now this turns out to be the covariance matrix of the filtered signal and the covariance matrix of the broadband signal. So we can rewrite this to be S and rewrite this to be R. I choose these letters because S is for the signal that we want to enhance, and R is for a reference that we want to enhance against. So what we are doing here is trying to identify a vector of weights, which we treat as a spatial filter that maximizes this ratio here, which is sometimes called the generalized Rayleigh coefficient. It can also be rewritten using this notation. Okay, now this doesn't actually tell us how we get the W's. This just re-expresses this form into a slightly different picture. So it turns out that the solution to finding the W, the weight vector that maximizes this ratio here, and by the way, it's very slight abuse of mathematical notation, but this indicates that when we find our W max, we can plug in W max to this equation. This whole thing boils down to a single number, so one scalar, which reflects the power ratio, and we can call that lambda. Anyway, the solution comes from considering not a single vector W, but instead a matrix of solutions W. So all these vectors turn into matrices, and we can also apply a little bit of linear algebra here. And we get to an expression like this, which turns out to be an eigenvalue decomposition on the matrix product R inverse times S. And then, you can also put the R on this side instead of worrying about the explicitly computing the inverse of R, which we don't actually need to do. So this is called the generalized eigendecomposition. Here you see some results in a validation study. So here I simulated the data, so I know what the ground truth result is. The red dashed line here shows the simulated result. So this is the ground truth, and the black and the purple outline show the result of the method. So you can see that this method, this algorithm JetBounds, does a really good job at recovering the ground truth data. Now it's a little bit smeared due to the leakage from the spectral filter. In principle, we could, I could have made that a little bit tighter and gotten this a little bit closer. But actually, in real neural data, particularly in EEG data, it is beneficial to add a little bit of spectral smoothing. So this is a pretty realistic scenario. Up here in the top row, you see the ground truth from the simulated data. Here you see the empirical maps resulting from the result of the JetBounds algorithm. And this shows the actual eigenvectors, which are more spatially selective compared to the component maps here. So it works quite well in data simulations, but on the other hand, we can always create the simulations to have low noise and high signal energy and get the results to work. Here you see some results in empirical data. So this is a resting state EEG data set where we don't actually know the ground truth, but these results do look pretty sensible. So we see some very low frequency components which appear to be blink artifacts or eye movement artifacts. Here we see something that looks like theta. This is the alpha component, maybe a second alpha component in a slightly higher frequency, and so on. I also applied this analysis method to a group of Parkinson's patients and matched controls. In the interest of time, I think I'm not going to go into detail about this, but you can read more about this in the paper if you are curious. So, in conclusion, spatial temporal structure in multi-channel data can be leveraged to identify components in the brain which we interpret as reflecting statistical sources in the EEG data, in the multivariate data. These decompositions or multivariate decompositions are advantageous over single trial analyses due to source mixing and also due to reduced signal to noise characteristics at the single channel level. I think this is a nice empirical method. It can help us identify individual variation in brain oscillation frequency and other characteristics, including the bandwidth. And this method in principle works for any multivariate data sets. I'm not showing this here, but I've also tried this method on data from simultaneously recorded LFP and EEG signals, and it also works quite well in mixed scale data.
Hello, I'm Jeff Schall. I'm happy to have this opportunity to share recent work that was accomplished in collaboration with a number of talented colleagues. Today's talk will feature work by Beatrice Herrera, Amir Sajjad, Jake Westerberg, and with Jorge Riera. Our lab obtained simultaneous recordings of EEG and laminar spiking with local field potentials in two areas of the macaque brain. First was in visual area V4 in monkeys doing an attention task that elicits the ERP component known as the N2PC. Other work recorded in a medial frontal area called supplementary eye field in monkeys doing a stop signal task in which an ERP component known as the error-related negativity is observed. With these data, we will address these questions today. First, how do multiple cortical areas contribute to a particular EEG component? Second, will biophysical models developed for granular cortical areas apply to agranular areas? Finally, how does oscillatory activity across cortical layers relate to EEG biomarkers? So, to use the macaque as an animal model of ERP components, we must first verify that homologous components can be found. Some years ago, Jeff Woodman began recording EEG from the cranial surface of macaque monkeys while they performed a visual search task. And as illustrated here, a component identified as the N2PC is observed in the monkeys. Inverse modeling of the voltages recorded over the cranial surface locates current sources in occipital and caudal temporal lobe areas. Other work in monkeys performing a saccade stop signal task identified the error-related negativity occurring after errors in this task. Inverse solution locates the current sources in medial frontal cortex, supplementary eye field on top and stretching down to anterior cingulate cortex. Parallel data collected by Jeff Woodman's lab arrives at a similar picture in humans. Now, linear electrode arrays were inserted into V4 on the prelinate gyrus of monkeys as they performed the visual search task. We ensured that the penetrations were perpendicular to the cortical layers by examining the progression of receptive fields across depth and the characteristic flash-evoked current source density in individual and on average across multiple sessions. In other work, linear electrode arrays were inserted into supplementary eye field on the dorsomedial convexity. We ensured that the array was inserted perpendicular to the layers through CT-MR imaging guidance and the consistent distribution of gamma power and spike isolation. We also observed a consistent but weaker visual flash evoked current source density in supplementary eye field. With these data, we can determine what current differences arise during different task conditions. This slide shows current source density on trials when a target was in the receptive field of the recorded region and when a distractor was in the region and the attention was allocated on the opposite hemifield. The current density, from the current density, we could derive the current dipole moment. That's plotted in B. Solid line are trials when the target's contralateral, and dotted line shows the current dipole moment when the target was ipsilateral. And that difference in the plot is the N2PC. We constructed a boundary element model of one of the monkey's head and brain and could position the dipole in the prelunate gyrus where the sample was recorded and do forward modeling of the N2PC voltage distribution. As shown on the bottom, in trials when the target was in the receptive field, when a distractor was in the receptive field, and their difference. And the difference plot results in a voltage distribution on the head that resembles or recapitulates the N2PC. Other areas in the monkey's brain and our brain contribute to the process indexed by the N2PC of target selection and attention allocation. This illustrates data recorded some years ago. Simultaneous recordings in frontal eye field of monkeys that isolated spikes and local field potentials were sampled simultaneously with the EEG over the back of the head, from which the N2PC was derived. So the N2PC that indexes attention allocation in this case arose after about 180 milliseconds. The local field potential in frontal eye field distinguished target from distractor after, say, 160 milliseconds. But the spiking rate of frontal eye field neurons distinguished target from distractor closer to 100 milliseconds after the array was presented. So frontal eye field signals precede in time the N2PC. With these data, we also quantified the correlation and fluctuations of both spike rate and local field potential voltage in correlated with the EEG. Spike rate didn't correlate with EEG. That's not surprising. But as illustrated here, across most sessions, there was consistent and pronounced correlation in the trial-wise fluctuations of the LFP voltage in frontal eye field and the EEG polarization recorded over the back of the head. These results indicate that the process indexed by the N2PC happens earlier in the frontal lobe, which can influence the N2PC generator. Now we were interested to see what voltage distribution is contributed in a biophysical sense from the frontal eye field. So in the same boundary element model, we placed the dipole in frontal eye field in the rostral bank of the arcuate sulcus. And the resulting voltage distributions, heat maps are shown to the right for target in receptive field, distractor in receptive field, and their difference. And this doesn't look like the N2PC. This indicates that while FEF contributes computationally to the process indexed by the N2PC, it contributes little biophysically to the observed voltage distribution described as the N2PC. Now transition. We've talked about V4 and frontal eye field, which are both granular areas of the cortex. We now want to compare supplementary eye field, which is agranular, to primary visual cortex, which is strikingly granular. In sensory areas, research over decades now has arrived at the view of a canonical cortical microcircuit, which is indicated on the left, which signals flow from the inputs to the granular layer, to the upper layers, down to the infragranular layers. The histological sections on the top are taken from primary visual cortex, and the laminated structure is clearly evident and characteristic. The histological sections on the bottom are from supplementary eye field, and the absence of layer 4 is clear, and the weaker and different laminar organization is equally clear. So we were interested to compare the physiology of these two areas. So I'm going to present two results, and both results lead to the conclusion that there are some similarities but many differences between granular and agranular cortex. First, we simply compare the flash-evoked visual current source density. In V1, current density, in V1, the initial sync was in layer 4, and then current sync spread to the upper layers, followed by the deeper infragranular layers. In supplementary eye field, the flash-evoked current density was weaker, but had characteristic structure. There is no layer 4. The earliest syncs in these data were in layer 3 and layer 5, and then layer 6, and then followed by layer 2, simultaneously or later. So there's similarities, but there's differences. A more rigorous test involved an analysis of cross-frequency phase coupling across layers. Earlier work that Alex Mayer, colleague at Vanderbilt, had shown that gamma power in the upper supragranular layers of V1 is synchronized or coupled with alpha phase in layer 5, so cross-laminar phase coupling. We replicated that result in V1 of 2 monkeys, and then submitted the supplementary eye field data to the same analysis. And as you can see, the phase amplitude coupling is much weaker and in supplementary eye field relative to V1. This is summarizing the data from 2 monkeys V1, 2 monkeys supplementary eye field. The matrices show Torts modulation index averaged across depth for the alpha phase signal on the abscissa and the gamma power on the ordinate. In both monkeys in V1, there was meaningful, significant coupling of gamma power in layer 2-3 to alpha phase in layer 5. That cross-laminar coupling was absent in supplementary eye field. What difference this makes has yet to be determined. Now, another characteristic feature of supplementary eye field is the existence of neurons that signal when monkeys make errors in the stop signal task. The spike density functions at the top plot the activity on error trials and on correct trials, and the difference is clear. The time depth plot at the bottom is illustrating the depth at which each error neuron was sampled and the duration of the error signal produced by that neuron. The triangles are broad spikes and the stars are narrow spiking neurons. Now, error processing has been described as a process of detecting the coincidence or the difference between what you meant to do and what you did. And a number of researchers, including Michael Cohen, have supposed that a cellular mechanism that could accomplish error detection and conflict detection involves comparing signals in apical and basal dendrites of pyramidal cells. And Larkham's work indicates the importance of backpropagating calcium spikes in these apical dendrites. So to explore this in our data, Beatriz Herrera, with guidance of Jorge, and we were along for the ride, developed a 2-compartment model of a layer 5 pyramidal cell that simulates the calcium spikes in the apical dendrites. Now, because it's just 2 compartments, it's a tractable model, so it is computationally possible to simulate thousands of these pyramidal neurons. The combined calcium spikes in thousands of these neurons produces current density of a particular form, and that current density simulated from the calcium spikes has many features in common with the observed current density from supplementary eye field of the monkeys during the stop signal task. Beatrice embedded 1,000 pyramidal cells in supplementary eye field on the left hemisphere and on the right hemisphere, and simulated the calcium spikes. And what's illustrated is the resulting voltage distribution on correct and error and the difference of the two. This voltage distribution resembles the ERN, and I'm showing the current density inverse solution to remind you of that appearance. So supplementary eye field positively contributes to the error-related negativity. Now, error processing has also been identified with enhanced theta power. And so we've begun looking at cross-frequency coupling between theta amplitude, theta power, and theta phase, and also gamma power and theta phase. On error trials, we observed enhanced theta power coupled with theta phase more or less uniformly across layers after errors. But gamma power coupled to theta phase showed negligible differences across the trial types. Finally, we were interested in the coupling of spiking activity and the rhythmic LFP. So we used the pairwise phase consistency measure. We found that after errors, pairwise phase consistency was enhanced at theta frequencies, at low frequencies, but only in layer 3 and in layer 5. These data recorded from V4 in monkeys doing an attention task and in supplementary eye field, medial frontal cortex of monkeys doing a stop signal task support the following conclusions. First, cortical areas can contribute computationally, but not biophysically, to EEG components. Biophysical models must distinguish agranular from granular areas. And finally, oscillatory activity and coupling varies with task conditions and across cortical layers. Thank you for your attention.